Uh, Dr. Srinivas is currently at School of Biological Sciences, NICER, Bhubaneswar. He received his PhD degree from the Indian Institute of Science, Bangalore in the year 2006. He carried out his postdoctoral work with Professor Mohan Balasubramanian at TEMSIC Life Science Laboratory and the Mechanobiology Institute, Singapore. He initially joined IIT Indore in the year 2014 and later moved to NICER in October 2015. His research interest lies in studying the dynamics of DNA segregation and cell division in the context of bacterial cells. And he primarily works on FTZ protein, which is an ancestral tubulin protein in bacterial cells. So for participants, please switch off your audio and video at all times. If you have any questions, kindly raise your hands or type in the chat box and we will relay it to Dr. Srinivas as and when he is ready to take the questions. So over to you, Dr. Srinivas.
Yeah, okay. Thank you so much for this introduction, and thanks the organizers for this wonderful meeting. And I think the sessions are going very well. I did attend a few talks, and there is some very good talks spanning across, like from the single cell level to the entire organism. So I think it has been a wonderful conference. So without delaying much, I'll just get started with what I'm going to present today. So as already introduced, so we are interested in studying bacterial cell division and DNA segregation in bacteria. And of course, as most of you know, it dates back to exactly 300 years before I was born, when Antony von Leeuwenhoek in 1676, in his famous letter, using his microscope, he had described this small things that he had observed in his water lens-based microscope called animal cubes. And since then, of course, we have come very far with what we know about bacteria and what bacteria are, with the Burgess manual in between classifying bacteria into several groups and all kinds of species. Probably this will be one of the biggest manual which segregates so much of classification that is being done and taxonomy that is being done. And but then, even though there is so much of diversity in this, what stayed with us is something which we all know as the workhorse, primarily because it divides rapidly. But what I'm not showing you the workhorse really, but something very similar to this. So there's an anaerobic bacterium which is Clostridium novii, and you would just see like just something is born out of an egg. One of these small little things that you see here are spores. Those will just crack open, and one of them will start growing and dividing. Yeah, so one of them cracked open, and then you have this bacteria. They're just within overnight they have taken over the entire microscope field. So this is the kind of thing that has alluded people to work on bacteria simply because they're easy, fast, overnight culture. You get E. coli, you can study them and those. But what it has led to is, in this famous paper when Roger Stanier formulated this concept of a bacterium, they declared something very bold for that time even. So what they said is there are only two kinds of organizations in biology. One that you see probably on the screen right now on the left-hand side, that is the eukaryotes that we all know about, the human cells and the fantastic the microtubules that is going from the nucleus to the cell ends, carrying all the motors, the actin fibers, the intermediary filaments, which all keep the cell in perfect order and perfect control right from the central organizing administrative wing. And it's all in perfect order. And all these are doing their job at the right time and the correct time and everything. And the second kind of organization that Roger Stanier put forth for the bacterium was essentially this. Essentially nothing. So the bacteria were not organized, they had nothing in them. It was just a bunch of enzymes that were there and a bag and a wall and a membrane that was always there. So this was the concept of the bacterium that weaned our ideas of bacteria. And including the cell division process that I just showed you was thought to be as simple as or as complex as a soap bubble, that you just stretch the soap bubble and eventually it will just snap. And then that's binary fission, and that's where the word comes from. That binary fission, it will just snap into two. And bacterial cell division was as complex as this process. And this idea was despite the fact that famous biologists like Francois Jacob, whom we all know for the Lacoperon, along with Hiroto, had identified the first ever cell cycle mutants in life forms. So this was way back in 1966, almost 10 years before Nurse and Hartwell had isolated the mutants. So despite this entire wealth of cell cycle mutants, the idea of bacteria being just dividing like a soap bubble stayed on for a very long time. And this is our human aspiration to see things. It was proposed several times that FTSG has a GTPase function that might be involved in cytokinesis, but it was not until late 1990. And this was, I think I was into the second year of PhD that this structure of this papers came out. That tubulin and FTSG were shown to be structurally very similar. And it is not until we actually see these things that we are very convinced. So that's the power of seeing things and this power of structural biology here. And not very late, just 2 years later or 3 years later, it was shown that MREB, another actin, like another protein in bacteria which is involved in cell wall synthesis, was exactly like actin fold. So it became very clear that the tubulin and actin, the cytoskeletal edifice of our eukaryotic cells, was actually originated in prokaryotes after these structures became available. So that really took us much further, and fantastic work from several labs across the world have really expanded the scope in a very short period. Like nearly after 100 years of denial that bacteria does not have a cytoskeleton, in 1990 or 2000, close to 2000, like FTSG was supposed to be the cytoskeleton, proposed to be cytoskeleton. A few more were discovered in the following years, and then now it's a huge family of cytoskeletal proteins and bacteria. So and so that we know that they are well organized inside a cell structure. So the brown structure that you see there, only thing is I don't know how a cursor works in this. I'm really sorry about this with using this iPad and with the keynote, I have not been able to manage a cursor, but it's labeled there. So tubulin is the homologue of it. That is the FTSG homologue that forms a cytokinetic ring. Actin homologues are varied in nature. One of them shown here in green is this that is pushing the plasmids apart and DNA involved in DNA segregation. There is another actin which is shown as a spiral here, which is, I think, from current new data, it is not a spiral organized structure, but it's still kind of lays the cell wall synthesis in these hoops or loops, and that controls the cell wall synthesis and morphology of the bacterial cell. And there are intermediate filaments that gives a curvature and several other functions. So what we are interested in our lab is studying two aspects of this. As one is the partitioning of the cytoplasm, which is the cytokinesis, and then is the partitioning of the DNA or the genetic material itself. So these are the two aspects that we work on. So I'll just start off with the second part here, which is the partitioning of the DNA first. So again, we cannot start this topic without I mentioning Franco Jacob here, because he was the first one to have put forth a formal model for how bacteria might, or how DNA segregation might be happening. Of course, the eukaryotes, it was already very clear there was very organized structures that were pulling the new DNA, and the metaphase plate were clear at that time. But in bacteria, he had, based on certain EM observations, he had suggested that the nucleoid is tethered to the membrane. And as the bacterial cell elongates, this replicated nucleoid tethered to the membrane will simply move to the sides as the bacterial cell is elongating. And these are also pulled along with that, and they are just segregated with that physical force. And since then, there have been a lot of molecules that have been identified and studied, and so it doesn't turn out to be so simple as what Jacob had proposed. So one of the most studied is this ALP, or the PAR-MRC complex, which is an actin-like protein in bacteria. So these are involved in DNA segregation, and Dyke Millen's lab and Ian Love's lab have worked extensively on this, and just a summary of this is shown in this small piece of video here. So what you see here is a DNA, which is drawn in the pink, and then there is an adapter protein that binds to a specific sequence in this DNA sequence, which is exactly like how kinetochores bind to the centromere in eukaryotic cells. And then there is an energy deriving protein, which is like the tubulin or the polymer forming protein, which would now help in pushing these plasmids apart. So what you can see here is this is a R100 plasmid. there is the sequence called as par C on the DNA, and par R is bound to it. And the actin-like protein, when it initially polymerizes, undergoes dynamic instability like the eukaryotic tubules. That is, binding ATP, and then hydrolysis leads to depolymerization. As soon as there are two plasmids, insertional polymerization sets in just like an actin, like the formants help it, and the polymers grow in length, and the plasmid is pushed apart to the cell end. So just playing it again. So this is the dynamic instability step that is like the tubulin in the eukaryotes. They depolymerize and polymerize. But then there are, when the plasmid replicates and there are two copies of them, they bind on, they form bipolar filaments, and insertional polymerization happens here, like the formin is allowing the polymerization. And the two plasmids are pushed aside to the cell poles. And the cell division would happen only in the middle, and the plasmids, each daughter cell would get a plasmid. And typically, this is the form of DNA segregation or plasmid segregation genes that is involved. You just need these three components. The cis element, which is the par C here, which is depicted in the pink color. The par R, which is the polymerizing protein itself, which is the adapter protein that binds to par C. And par M, which recognizes par R and forms the polymeric structures. So there are several actin-like proteins, and we had earlier worked on some of these EM structures. So you get a variety of helicity in these structures, enormous diversity. Unlike the actin, which is plain 166 degrees turn, you have an enormous diversity of actin-like proteins and bacteria functioning in different species. And each of them contribute to plasmid segregation in their own way and have different properties. Some form tubes, some are just opposite filaments, some are parallel filaments, some are helical filaments, but all of them essentially do the same function. Bind the DNA, push the plasmids apart, and they help in plasmid DNA segregation. But one among them is a very unique class in bacterial, which has come to be known as the Walker A class of ATPases or cytoskeletal ATPases. This class is very unique to the bacterial family or the bacterial kingdom. These cytoskeletal elements are belong to the large family of the P-loop ATPases, which we all know about. So these are just a subset of them, then they're known as the par A or the MIN-D family of proteins. And for this particular protein, several models have been proposed in the past, how they segregate plasmids. Unlike the actin, which I just showed you, it is not clear how exactly this Walker A class of ATPases function in segregating DNA. And while the actin that I just showed you is present in plasmid DNA largely, it is this Walker A class of ATPases which is found in genomic DNA segregation in bacteria, including the famous bacterium, which is the Vibrio cholerae, one of the pathogens, Pseudomonas aeruginosa, mycobacteria. So most of any pathogen you take, most of them tend to have this Walker A kind of ATPase and they rely on them for DNA segregation. So how do these function? Several models have been proposed. I start with this oscillation foci. People found that there are these foci and that go back and forth between the two poles of the bacterium, and eventually probably there'll be two copies. And when the cell divides, there'll be two copies inherited by the daughter cells. And then there was this famous burn bridge model that was proposed, just like the kinetochore. When it is depolymerizing, it pulls the chromosome. So a pulling model was proposed, and a filament pushing model. And more recently, there have been two different models, which is the diffusion ratchet model, the DNA relay model, or the hitchhiking model. These three models are not exclusive to each other. They probably operate in an overlapping manner that each of these components plays some role. So the DNA relay model, the elasticity of the DNA becomes important for movement of these proteins and pulling the DNA and segregation of the DNA itself. And I'll just come to the diffusion ratchet model in a moment. And hitchhiking model is that within this bacterial nucleoid, these proteins which are involved in the segregation are embedded, and they kind of go along with the DNA, and which is in some kind of sync with the DNA relay model. So they are able to do the DNA segregation by organizing themselves on the nucleus. So all these three models, what it essentially says is that the segregating protein, which is this Walker A kind of cytoskeletal ATPases, which in this plasmid called as fertility plasmid or the F plasmid we all know about, is called as swapping. So this protein is a Walker A ATPase, as I told you. It can bind DNA, and more recent evidence has shown that the protein itself is localized mostly on the bacterial nucleus. So then giving way to the other models like the polymerization models, and those have gone out of fashion, or there is no support for those models currently. So the currently accepted model, which is the diffusion ratchet model, is simply goes like this. And these are again from experiments that were conducted in vitro very elegantly by Bacterial et al. Some time back. So they had a glass slide where the DNA was coated, and the bead that replicated that kind of represented the plasmid with the SOP-B protein coated on it. And with magnetic tweezers, they can hold it close to the DNA, close to the cover slip. And the model goes something like this: that you have two plasmids here, and the light green ball that you see is the ADP bond form. So they cannot bind the nucleoid surface. But the plasmid DNA, I'll just play this again. Yeah. So the dark green is the nucleoid where the Walker-A ATPases are bound. And when the SOP-B, this adapter protein, binds to that, it stimulates its ATP hydrolysis, releases it, thus generating a gradient across the chromosome, and the plasmid eventually moves across the ATP gradient. So this is the diffusion ratchet model that has been proposed, and is a well accepted. There is sufficient evidence that this is what is happening in bacterial chromosomes and the SPARA mediated DNA segregation elements. But what struck us is that there were some older studies that alluded to the earlier thing, what I said, that Jacob and Menard had proposed, that there is a membrane binding aspect to this DNA. So earlier papers had reported the SOP-A was also membrane anchored, and it was also found in membrane fractions in bacteria. And the plasmid was also found in certain mutants like the MUC-B mutant, which is an FMC equivalent in bacterial cells. So they are required to maintain compact nucleoid. Without them, what happens is you get a lot of anucleoid cells. So if this entire segregation process was dependent on the nucleoid, you would not have expected to find plasmids in anucleoid cells, because the plasmid is closely associated with the nucleoid. But the F plasmid and P1 plasmids have been shown to be in anucleoid cells in these kind of in these MUC-B mutants. And given that they also anchor to membrane, we thought probably there is some membrane binding aspect to these, and we wanted to explore what this is to just start with, whether this is true and what might be the membrane binding contributors or what role it might be playing. So what we found is that there is an amphipathic helix at the very C terminus of this particular protein SOP-A, which is absent in the P1 part. And this amphipathic helix, as you know, that it's mostly responsible for tethering any protein to the membranes. And this definitely has an hydrophobic phase. So like in other membrane proteins such as FTSA and MIND, which are bacterial cell division proteins that have been shown to associate to membrane, you see a very clear hydrophobic phase. Even on SOP-A, you see such very clear hydrophobic phase, suggesting that this might actually be leading to a kind of membrane localization. So, and in silico, we just tested this is in collaboration with Anand at IISc at MBU. One of the master students carried out this work in collaboration with him. And in silico, we can see that this peptide probably has some affinity to membranes, and it can go and embed on the membrane surfaces. It doesn't penetrate the membrane as expected of an amphipathic helix, but it can associate with membrane surfaces. Not only that, we could also show that by disrupting the the PMF or the proton motive force of membrane, the localization of SOP-A could be affected. So it's sensitive to the membrane potential of the membrane. So most of the membrane proteins do have this phenomenon that they show sensitivity to localization upon perturbing their membrane potential. So finally, we do find them again as reported that they are in the membrane fractions, and mutation in these residues or hydrophobic residues here cause the plasmid loss. So these residues are also important for maintaining the plasmid in E. coli cells. So there was something to membrane binding which we are still exploring, but what we don't know is if this region is sufficient for membrane binding in biological cells, or is it really membrane bound, or amphipathic helices are also involved in maintaining protein structures, are they involved in different functions, and this is not related to membrane binding. We still are exploring those things. We still do not know exactly what this region is and whether it is sufficient for membrane binding. So, however, during this course of studies, what we found is there was one particular mutant, this W362E here, which formed cytoplasmic filaments. And this was interesting because there was yet another mutant already published in the literature, which is the SOP-A1 here in the second panel, which also formed the cytoplasmic filaments. So one aspect, what are these cytoplasmic filaments? And in relation to, if I just go back to this slides in FTSA, mutating this one of these W's in FTSA results in exactly similar cytoplasmic filaments and disrupting the membrane localization. So we thought maybe this is related to membrane localization, or maybe may not be, but still we wanted to explore what these polymers are. So these polymers can be shown in E. coli, and we also expressed in heterologous system in a yeast system, and show that these are independent of the bacterial system, that we can get these proteins tend to polymerize by themselves. So somehow stabilizing the polymerizing phenomenon of SOP-A, which is known to polymerize in vitro. In vivo, earlier studies had suggested that there may be polymers, but later there was no evidence for polymers. But somehow these mutations might be stabilizing these polymers, and that is why you end up with this long cytoplasmic long-range polymers. So are these polymers really because of SOP-A that we tested by using an ATP hydrolysis mutant? So if we made an AK120E which does not bind ATP, you do not find any more polymers. So essentially these polymers are indeed dependent upon SOP-A binding to ATP at least. And without ATP binding, they are completely they are diffuse in the cytoplasm, and they do not show any kind of polymeric structures. But what was surprising is these polymeric structures were not bound to the nucleus. It had affected the nucleoid binding. And to demonstrate this very clearly, what we did is we simply treated cells with cephalexin and condensed the nucleoid with chloramphenicol. Then you can see more internuclear spaces. On the topmost panel, you see the wild type SOP-A, which is completely co-localized with the nucleoid. But these filaments tend to be not associated with the DNA. So this we can also show by using MinB strain, which forms anucleated mini cells. So these yellow arrowheads that you see in the bottom panel are cells which do not have DNA, and there are small mini cells because of polar divisions in bacteria. So these polymers are also found in such mini cells, showing that these polymers are not bound to DNA. So, but then why are these polymers not bound to DNA? What is causing this polymer? Is the polymerization stopping them, or what is causing the SOP-A not to bind a DNA? Was the next question that was. So, but this a small introduction is given this. So what is known about SOP-A is that when SOP-A binds ATP, it is still incompetent to bind to DNA. It is only when SOP-A is actually converted after binding ATP, it goes a conformational change. It becomes competent enough to bind the nucleus. So this in the literature is known as SOP-A ATP star state, and which is being regulated by one of these Walker A motifs, lysine, which is again pointed in green here. This is the lysine 120. So this lysine residue plays a very important role in controlling this conformational change, and different mutations in this lysine give rise to different phenotypes. So what we did is we mutated this K, again, this is the same K as the K120E. So we mutated these to different amino acids and checked if which of these sustain polymerization, or which of these can maintain the polymers of SOP-A. So what we see is neither R nor Q could maintain the polymers, but it was only Ka which maintained the polymer, which still could polymerize. So this is in combination with W362E and K120A. So only the Ka mutant was competent enough for polymerization, but the R and Q were not. So again, the ATPase activity of Q is equivalent to that of wild type. So this polymerization is not dictated by the ATPase activity, but it has to do more with probably this conformational change upon ATP binding to this ATP star state. So what we infer from this is that the Kr and Kq do not undergo conformational change, but the Ka is still able to undergo this conformational change to the ATP star state, which is therefore allowing a stable polymerization. So indeed, the polymerization also, like DNA binding, is dependent upon this conformational change. So, but if this is true, if Ka is the one alone able to undergo this conformational change, but not Kr and Q, then in the presence of SOP-BC complex, what one should see is Ka should behave like wild type, and the other mutations will be different from the wild type. So this is indeed what we see, that the wild type forms this foci in the presence of the SOP-BC complex within the bacterial cell on the nucleoid. And it is only the Ka which forms these foci, and none of the other K mutants can form the foci. Again, reiterating the idea that Ka is the mutation that allows the conformational change, but K to E, R, and Q block this conformational change. And therefore, the polymerization also is dependent upon this conformational change to ATP start state. So the next thing that we asked is if these are not binding non-specific DNA, have they just lost their DNA binding activity, or do they still maintain some DNA binding activity? So the SOP-A protein has two kinds of DNA binding activity. One is the non-specific DNA activity, non-specific DNA binding that it makes it localized to the chromosome or the nucleoid. The other, it also binds to its own promoter to suppress the transcription and acts as a repressor, which is again aided by SOP-B complex. So if the and very recently, like last year December, it has been shown that if the SOP-A protein is unable to bind to the non-specific DNA, but it is capable of binding to the specific promoter, then it functions as a super repressor. And what do I mean by super repressor? Is simply that even without the SOP-BC complex, these can act to repress transcription from its own promoter. So we tested this W and Q mutants which form this polymers for this effect of repression. And you can see even without the SOP-BC complex, which is on the left-hand side of the graph for the beta-galactosidase activity, they show very high level of repression, which is not again influenced by SOP-BC. However, this is not true for wild type. The wild type shows very weak repression in the absence of SOP-BC, which is significantly enhanced when you have the SOP-BC complex. So these two mutants do not bind non-specific DNA, but they are fully capable of binding their own promoter and repressing their transcriptional activity. So what we think is going on here is in case of wild type, the polymerization is not very strong enough that we have not detected polymerization so far, except for in vitro upon ATP addition. In vivo, there have been no polymers that is detected. The ATP star state allows it to bind to the nucleoid, but in these two mutants, there is some kind of conformational shift in the C-terminal helix, which allows the polymers to be highly stabilized, and therefore we can see these very stable polymers. And this polymerization is indeed dependent upon ATP binding and also upon the ATP star state conformational change. If we block that, the polymerization is also blocked. And of course, having formed the polymers themselves somehow have an orientation that they cannot bind the nucleoid, or the sites are blocked in this polymers for non-specific DNA binding activity, but not the specific DNA binding activity that is controlled by a completely independent domain within this protein. So that's what we think is happening with these mutants. So, but still we need to, I think a lot of biochemical study and a pure biochemistry study will help us clearly tell what is happening with these mutants and what stage it is blocked, and how this polymerization and the DNA binding are being regulated in this SOP protein. So from here on, I'll just switch gears to talk about the second topic, which our lab is interested on, which is the bacterial cell division of cytoskeleton. But in that, what I'm going to talk about today is using Fission Yeast as a platform for screening small molecule inhibitors for bacterial cytoskeleton, especially the FESZ protein or the bacterial cell division protein, which in the literature has come up as a very good target for antibacterials or new antibacterials. So what we had done a few years back when I was a postdoc back in Mohan's lab is that we studied a lot of these bacterial cytoskeletal proteins expression in yeast cells, and express like all the actins, tubulins, intermediary filaments, and metabolic enzymes like CTP synthase, which forms polymers, Walker ATPases, archaeal proteins like escort. All of them form polymers in this yeast, and we have kind of established the yeast as a good system to study the dynamics of these proteins independent of their bacterial interacting partners and whatever may be happening with the bacterial cells. And so in this regard, we had to expect nearly 40 different actins. All of them form polymers. So a few are shown here, and including the MREB, which I talked about in the introduction, that assembles the cell wall synthesis in the rod-shaped bacteria, the tubulin, which is the FTSZ, which assembles into ring-like structures, and another tubulin that is involved in DNA segregation in the pathogen Bacillus anthracis. All of this we had expressed in yeast system and studied the dynamics and polymerization properties of these. So the yeast system proved a useful model back then to study the polymerization of these proteins, which was difficult in bacteria then. But then what it also turns out is that this also seems to be an interesting system to identify small molecules that can probably affect the bacterial cytoskeleton. So what we had shown back then again is a small molecule A22, which is a known inhibitor of MREB. When added to the yeast cells, it prevented the polymerization. But if you add A22 after the polymers are formed, it would do nothing to them. So based on that, we had proposed that A22 was a competitive inhibitor of ATP binding, and probably it aided to prevent polymerization. So further moving on from this, we thought we should also try and establish this as a model system to screen for antibacterials, and can we use this for FTSZ as well? So what is FTSZ? As I told you, it is also introduced to you in the beginning that it is the bacterial cell division protein, which is the tubulin homologue. In eukaryotes, the cell division is done by actin, but in bacteria, it is this tubulin homologue. And this is not just bacteria, but chloroplasts have utilized FTSZ to divide. And mitochondria or ancient mitochondria also use FTSZ to divide their mitochondria. And eventually, by the time eukaryotes evolved, FTSZ has been lost from mitochondria in these eukaryotes, and dynamins have taken over. And in chloroplast, dynamins are in sync with FTSZ. So FTSZ is the bacterial homologue that was doing the cytokinetic functions. It forms a ring-like structure in the midcell where the cell wall synthesis. And it's very close to tubulin. It forms polymers just like tubulin. Of course, it's a single protofilament of tubulin, not like a tubule structure of tubulin. The GTP binding pocket is very conserved. The hydrolysis loop is highly conserved. There's a second subunit. The top subunit loop is the one that triggers the hydrolysis in both tubulin from the beta tubulin and in FTSZ. So these are very highly conserved structures. And in yeast, we had expressed some time back and shown that FTSZ assembles into ring-like structures independent of any other structures or any bacterial proteins. So it's kind of a self-assembly property of FTSZ. And Erickson around the same time had shown that FTSZ, purified FTSZ, when tethered to membrane, can assemble into ring-like structures. So given these FTSZ ring assemblies is a self-assembly process and it does not require any other proteins. It's a self-organization property of FTSZ to assemble into a ring-like structure. So what we had done back then again is how do these rings assemble? So one of these movies you can see on the left-hand side from the cable, somehow you go on to forming this spot-like structures. Of course, you cannot make out anything what is happening there because of the crowded nature of those dots and ring-like structures. So if you can see one of the mutants that we made, which has a slow GTP hydrolysis, it really captures how the ring assembles. So you can see this ring, the filament going on to a ring-like structure, it kind of spools into the ring-like structure. So the entire filament is kind of drawn into the ring-like structure, the ring is assembled. And this indeed is driven by a spooling process in this place. Yeah. You can see this ring almost like rotating and the tail being spooled into the ring. And by frap, we can show that the bleached region is actually moving towards the ring-like structure. That is shown on this right-hand side panel. So as time goes, the bleached structure is moving towards the ring and eventually gets into the ring-like structure. So this is we believe that FTSZ forms are assembled into ring. And today people have kind of validated this, that treadmilling in FTSZ is very important for ring assembly and ring construction. And very recently, there have been some really wonderful papers looking at doing imaging in bacteria. That FTSZ undergoes this kind of treadmilling process where the polymer keeps moving towards one end and depolymerizing from the other end. And that is what drives the entire ring assembly process. So, but since our idea was to see if we could establish a small molecule inhibitor screen for FTSZ, we were interested in pathogenic FTSZ species here, and not the E. coli. So we expressed Staphylococcus aureus FTSZ as well as an Helicobacter FTSZ in yeast cells. And you can see these again form like very nice ring-like structures. Of course, these ring structures were discernible or hardly discernible, I would say, even with super resolution microscopy, which is the 3D SIM here. This is structured elimination microscopy method used here. You can hardly see the SIM-like structures. And Helicobacter, interestingly, it forms helical polymers. Of course, Helicobacter and helical go well, but it has probably nothing to do with the Helicobacter's helical, but it is just some property of FTSG that it stabilized as a helical polymers when we express them at least in the yeast cells. So the drugs that we choose, first we wanted to establish the system. So therefore we just took these 3 drugs minimally to see what is the effect on FTSG. So of these, the 2 drugs, berberine and sanguinine, are known to affect several things, including eukaryotic cells and kinases in eukaryotes. Sanguinine affects microtubules in eukaryotes. But nonetheless, they have also been shown to bind FTSG and inhibit FTSG ring assembly. The only real valid drug in this case is this bottom left-hand side corner, which is the PC190723, for which the crystal structure has been solved with the FTSC crystal structure. We know the binding site, what are the contact sites and everything. So that's probably the only valid real FTSC drug that is available so far. And it did go up to clinical phase 2, but then for some reason it failed there. So we used these 3 drugs to see if they had any effect on FTSC polymerization of 3 different species, that is E. coli, staph, and Helicobacter. So of course, sanguinine and berberine did affect the FTSG assembly, as has been reported earlier, but we cannot really say if these are direct effects or these are indirect effects through the yeast cells, because these also tend to affect the yeast cells. Although we don't see any much defect at the concentrations that we use here, it's still hard to say whether the effect will be direct or indirect. But with PC, we can see that PCs acted to stabilize the FTSG polymers. As you increase the dose, you get more and more polymers, because PC is a drug that binds the, go back here, it binds this interdomain cleft. And therefore, what happens is it blocks this domain movement between these 2 structures, one the dark blue and the light blue here. And then it prevents the GTP hydrolysis from happening. So it stabilizes it in the polymeric state, and therefore what we get is this effect. That when you add PC, it is not inhibiting polymerization, but it stabilizes FTSG function, but that is also inhibiting. So if you stabilize the FTSG function, the cell will still not divide again. And that also is toxic to bacterial cells, and therefore there's still a drug. So either you depolymerize, or you can even you can have drugs that also stabilize its function. So in case of both Staphylococcus and Helicobacter, the PC190723 drug seems to stabilize the FTSG polymers. So you can really see when controllers does not have any polymers, the drug-treated cells have already got polymers, so suggesting that they have lowered the critical concentration of polymerization for FTSG. So we further go on to see, because since it blocks the GTP hydrolysis activity, we wanted to see if it also blocks the turnover rate, because the turnover rate is dependent upon GTP hydrolysis activity. So FTSG polymers are very dynamic. They hydrolyze GTP around 4 to 5 GTP molecules per minute. And so they also undergo turnover polymerization and depolymerization, which can be easily captured by this assay called as FRAP, which is the fluorescence recovery after photo bleaching, which you have heard in several talks during this course of this workshop itself. And so you can see in the first panel where there is this pre-bleach, and then after bleaching, what we have is a small recovery, and we can calculate the 50% recovery, which gives you the T half of recovery. When you treat cells with this drug PC190, the T half of FTSG really goes up in case of Staphylococcus aureus, and suggesting that the turnover rate is reduced, and the drug is actually acting to stabilize or block the GTP hydrolysis activity. And this is also true for Helicobacter pyrrole, that the recovery rate is slowed down, or the dynamicity is slowed down in case of FTSG polymers when you treat with this drug PC190723. So what was interesting is when we started this, we didn't expect Helicobacter pyrrole to be actually sensitive to PC. We thought that it would be resistant. We know staph is sensitive, so we just use Helicobacter as something that would be resistant, because you can see this red marked R residue there and a star there. So all the species which have this R there in that position, or a histidine like Pseudomonas aeruginosa, all of them have been shown to be resistant to PC. And it is only Staphylococcus aureus and Bacillus which have a valine in this position, very sensitive. And it has been therefore suggested that this arginine plays a critical role in resistance to PC. And if you have an arginine, it will be resistant. So that is how it was deemed. And these resistance studies, as you know, will be done by simple minimal inhibitory concentration. And the PC drug might not even enter Helicobacter pylori, and therefore it might still grow, and you will still conclude that it is resistant. But the resistance is not because of a mutation in FTLC here. It is because of something else. So we sought to see an explanation of why Helicobacter is actually sensitive to PC. So what we realized is it is not just this R, but if this R happens to make a salt bridge with another residue, like in case of E. coli, it makes a residue. This R307 makes a salt bridge with E198, shown in the left bottom panel here. So that is what blocks the PC binding site, the cleft, and the PC cannot even enter the cleft. And that is what makes it resistant. It is not simply the presence of this residue. And so in Helicobacter pylori, what happened is, although this residue is R, the equivalent of the E is a T216 here, which cannot engage in a salt bridge with this R. So therefore, there is no blocking of this binding site, and it could really penetrate the structure. And we have more evidence now to say that this T, if you restore the salt bridge, you can actually it actually results in a resistance in PC binding. So that we can again show by FRAP and by doing this qualitative counting that I just showed you. So we have those evidence, and we think that it is the salt bridge that actually leads to the resistance, and it is not the single residue in contact with PC that leads to the resistance. So in summary, what we have seen is that Staphylococcus and Helicobacter pylori are assembled into polymers in yeast. So this adds to the list of FTSZ polymers that we have shown that is capable of self-assembly in yeast. So it's really FTSZ has intrinsic property to assemble into ring-like structures, and PC stabilizes the FTSZ polymers even when expressed in fission yeast. And Helicobacter pylori FTSZ can probably bind to PC, and the resistance to PC is mediated by salt bridge interactions across the interdimensions. So that brings to the end of the talk, and I'd just like to mention the people who did this work. So the first part, the SOPE or the PARE part of the work on plasmid partitioning was carried out by Deepika mostly, and then Nivedita and another master student, Sakshi, in my lab, in collaboration with Anand's lab for doing the in silico membrane association and molecular dynamic studies, and also help from Gayatri on structural aspects of these proteins. And coming to the FTLC work was mainly carried out by Ajay and Sakshi. And the work was actually initiated by a master student, Bhagya, and later on taken over by Sakshi, and Ajay has taken forward to this kind of state. And Sakshi is working on the resistance aspects of those mutants, the FTLC mutants. And this again, the structural aspects which I showed you is the salt bridge giving the resistance is in collaboration with Gayatri's lab, and Joyita is carrying out that the structural aspect of the work, and the modeling work was done by Joyita at Gayatri's lab at ICERCOM. And it would just be not possible without generous funding from Department of Atomic Energy, and not just Atomic Energy, but here Department of Biotechnology and also DBT funded these projects and some of this work. So without their funding, none of this work would have been possible, and we have some very good facilities to carry out such microscopic studies with bacterial cells here. And you will see in the afternoon, like Nivedita and Sakshi will present some of the quantitative aspects of how we can go about quantitating FRAP and doing FRAP studies, and also how you can use certain parameters to actually quantitate the effect of the drug on small molecules on these cytoskeletal proteins. So with that, I would like to thank everybody and all the collaborators and all the people who are here, and I would be happy to take any questions.
There are some questions in the chat box.
Let me just see if I'm also.
I will speak it. So first question is, do these ring-like structures of FTZ once formed in yeast also disassemble on their own, or do they remain like rings?
So these structures are very dynamic structures, so they're constantly appearing and disappearing, but they do have some lifetimes. So if you do a FRAP on them, even within the ring, the structure is stable there, but the polymers are constantly turning over. So there is a constant assembly and disassembly, and eventually the ring itself has certain lifetimes. They don't stay forever as single ring, but it keeps appearing and disappearing. But overall, in the population in the cell, you will find a sufficient number of rings at any given point of time. But if you block the GTP hydrolysis, like what I showed you in one of the movies, which form this nice pooling structures, those rings, once they form, they don't disassemble because you have completely blocked the GTP hydrolysis.
Thank you, sir. The next question is, do you think actin binding proteins such as ARP23 complex are also involved in stabilizing the FTZ polymer bundles?
Yeah, so that's something which since 2007, I think, when I have been presenting this question, always comes up. And I tell, yeah, we must do a tap tag and we must find any interacting proteins which are there. But we haven't done that so far. But I used to give one answer to that. That is, Margolin, who had worked extensively on FTSE, had expressed these proteins in mammalian cells in the cytoplasm. They had done a pull down and they didn't find any interacting protein. But having said that, very recently, again, last year, what has happened, what they have done is Pascal Cossat's lab, which Ravi's group and all of you will know, is they have expressed FTSZ inside the mitochondria of human cells. And they have done pull down and they find a lot of interacting proteins with FTSZ. So it's not like FTSZ is probably not inert in the eukaryotic cells as we assume. Probably it's a good time to revisit and see if there are also cytoplasmic proteins that interact with FTSZ given the high sensitivity of mass specs that has happened over the last 10 years. So that is something I wouldn't say yes or no, but I would probably no and maybe some yes. But how much they influence the ring assembly and these things, that I doubt, because in vitro also we can show the similar dynamics even without any of these proteins.
Thank you, sir. The next question is whether this compound that you have identified could only be targeted against S. aureus or Helicobacter infections,
Oh, sorry.
or
Okay.
it might act as a broad spectrum.
Okay, so sorry. First thing, if I conveyed that I discovered this drug or we found this drug, I'm very, very sorry. As I showed you in the first slide, these are 3 known drugs that we wanted to establish the system. Therefore, we use the 3 drugs that have been highly established to show that they target FTSZ. So the drug was discovered by Errington's lab and synthesized in Errington's lab back in 2008. So we did not find any new molecule. What we wanted to establish is using the known molecules. We wanted to show, yes, this can be done in the yeast system. So if you want to do a screen, possibly drugs can be screened with the yeast system. That was the idea of these experiments. We have not gone to a stage where we actually made any small molecule discovery or taken it to a high throughput screening system. That is one question. Answer the first part. And the second thing is the PC is not a broad spectrum antibiotic. It was believed to target only gram-positive bacteria and not gram-negative bacteria. And within gram-negative bacteria, there had been a lot of controversy that it affects E. coli or it doesn't affect E. coli. And as we were doing this work, it was clear that it does not affect E. coli. And more recently, it has also become evident that PC does not affect E. coli FTSZ. But the problem with the gram-negative bacteria is that they have an outer envelope. And it is because of the outer envelope, the PC is not even penetrant to the bacterial cytoplasm, which is what we believe is happening with Helicobacter as well. Helicobacter also has an outer membrane which is even more tougher than E. coli, and it is not penetrating Helicobacter, but the FTSZ itself from gram-negative bacteria may be sensitive. So if you had a combination, why this is important? Because now, instead of thinking that no, gram-negative FTSZ is resistant to PC and gram-positive is sensitive, we have a different view now that yes, gram-positive FTSZ can also be sensitive to PC as long as you can get the drug inside. So if you have a combination or there are scaffolds that are being made that can make the membrane permeable, if you can couple PC to one such scaffolds and get it across the cell in a gram-negative bacteria, it can target FTSZ. So that's why this becomes important to understand the resistance mechanism. So because normally people get, how does it matter understanding resistance mechanism. How does it help in drug discovery? We just find drugs and that should be sufficient. So here is an example to say that yes, understanding mechanisms can change existing thinking and thought. So PC itself is, as of now, it's not a broad spectrum. It can only penetrate gram-positive cell walls and not the gram-negative outer membrane envelopes.
If anybody else has any more questions, they can unmute yourself and speak up. I think there are no more questions.
Okay. No more questions yet. Yeah. Okay.
Thank you so much, Dr.
Okay, then.
Srinivas, for joining us and for putting this presentation together. It was a really interesting talk, and we will start with the hands-on training by Nivedita and Sri Lakshmi at 3:30. We'll take a short break now.
Thank you,
Okay.
Shini.
Thank you so much. Yeah.
Thank you so much.
Thank you all. Yeah.
Nice talk.
Thank you.
Thank you
Yeah.
so much.
Thank you, sir.
Thank you. Thank you.